I'm gonna introduce today's seminar speaker, Dr. Rosalind Abbott. She's here from Tufts University, and she's interviewing for a faculty position in biomedical and mechanical engineering with a biomedical engineering emphasis. Rosalind got her BS and MS degree in biomedical engineering from RPI, and she received her PhD in bioengineering from the University of Vermont. And currently, she's a postdoctoral fellow in adjunct professor at Tufts University. She works in the laboratory of Dr. David Kaplan as a postdoctoral research is mainly focused on tissue engineering project for adipose tissue. And her future research plans are to focus on adipose tissue models to study diseases like obesity and diabetes.
Thank you so much for the intro. We're back on time today. I've been late all day, so this feels good. So I like to start with this slide because I think we all think about obesity as an ever-growing problem, but then we often don't think about how it really affects the progression and actually the initiation of so many other diseases. And really how an expansion in your adipose tissue can actually affect other tissues systemically in your body. So it's got a big, far-reaching sort of effect. So today I'd like to talk about some work that I've been working on in David Kaplan's laboratory on the development of a tissue engineering adipose model, and that's really for the specific purpose of studying metabolic diseases. So I thought I'd go through my journey so far. So I started out at RPI, as you mentioned, doing my bachelor's and master's there. And because I love skiing and I love snow, I went up to the University of Vermont and did my PhD there. In the last few years, I've been down at Tufts in Boston. So at RPI, I really got interested in how can we use model systems to answer meaningful questions. So really, how can we provide really defined boundary conditions to really drive our hypothesis-driven research. And whether that's in silico or in vitro or in vivo, and how we can kind of combine these different methods to control and define our perturbations more appropriately. So as an undergrad, I started off working for Dr. Brunsky at RPI, and we looked at implant interfaces with the tissue. So how micromotion affected the strain distributions there. So we had an in vivo model where we could use that defined boundary condition, and then we would take micro-CT images, and then I would import them into finite element software and analyze the strains. And we found that areas of low strain or high strain resulted in low matrix deposition. And so then he went on sabbatical, and I worked in another lab for my undergraduate and my master's, and that was more focused on the intervertebral disc. And so what we did was we called it the degenerator, and we put these two rods into the two vertebrae, so the intervertebral disc is between two vertebrae and the spine. And we used defined loading conditions to mimic larger than physiological loads. So we were thinking that the reason why truck drivers had a greater incidence of low back pain was because they were exposed to higher frequencies of load. So we used that system to really control the defined boundary conditions again to provide those mechanical inputs. And we found that those accelerated frequency rates actually caused accelerated degeneration. So then at the University of Vermont, I started to work on low back pain further. And so we used, in my first lab, I worked with Dr. Iatrides, and we looked at controlling the boundary conditions by using these alginate beads to put degenerated disc cells in. And I looked at different regenerative approaches that were based on development. So the notochord, which is how the IBD actually is created during development. And so he actually moved down to Mount Sinai, so I moved into a different lab. So he was very mechanics-based. And so I stayed actually working for both of them, but then I had a new advisor, and she was actually in the neurology department. And she was interested in acupuncture and how acupuncture changes the strains in your lower back and how that affects the cells and the matrix. So she was seeing this really interesting result where she'd take ex vivo tissue and she'd apply a mechanical strain on it, and she'd see the cells that are normally very fibroblast-like and very skinny, and they turn into these like large pancake-like cells. And that would control the viscoelastic behaviors of the tissue. So we were wondering, is this a matrix effect or is this a cellular effect? So what I did was I developed a collagen gel model where we could control the density of the matrix, because she was seeing in this loose connective tissue this large effect of the cells turning into these big pancakes. But then the adjacent dense connective tissue, they didn't have that response to stretch. And actually, as you develop low back pain, your tissue becomes more dense in your lower back. your loose connective tissue becomes more dense. So what we did was we put those cells that were obtained from the loose connective tissue and we put them in a dense matrix, and they didn't respond to stretch in the same way. We took those dense cells and we put them in a loose matrix, and they did respond. So it was really a matrix-dependent effect. Sorry. So this got me really interested in how we can make more complex models and how we can answer more meaningful questions. So at Tufts University, I decided to work for David Kaplan. He's a big silk biomaterial guy, so I really wanted to learn more about silk as a natural biomaterial. And then also I wanted to learn how to develop more complex human tissue engineer models because he's developed a lot of really complex co-culture systems in these 3D matrices. So you can't really see it very well, but that's a silk scaffold.
So we can adjust it.
Okay, so why do we want a white adipose tissue model? Really, there's two big reasons. The first one, oh, wonderful. And I can see all these people better. So the first one would be for a patient-specific human disease model. This is really because animal models often don't recapitulate the human situation. It's pretty well established that 2D models aren't consistent with 3D models. There's ethics with the testing components on humans, obviously. And also that the human population is so diverse. So it responds to things very differently. On the flip side of that, a white adipose tissue model is also really good for autologous soft tissue regeneration. So this is where you have a large defect, and whether that's from tumor resections or trauma or congenital abnormalities, you've got this large space that needs to be filled with adipose tissue. So current adipose tissue grafts and natural biomaterials alone, they actually resorb too quickly to maintain that tissue regeneration. So they lack the long-term stability required to provide that defect fill. They also lack the mechanical integrity. They lose volume over time, especially if it's just an adipose tissue graft on its own. And they often, especially with the natural biomaterials, will get these fibrotic capsules that will form right around the materials. So some white adipose tissue basics. I think we all kind of think about adipose tissue as maintaining our body contours for better or for worse, but also for mechanical protection for all of our different organs. But it's also a very active endocrine organ. So it secretes adipokines and cytokines that affect the entire body systemically. It stores excess energy, nucleocular lipid-filled vacuoles, which are these round staining right here of the adipocytes, and those are really stored with triglycerides. And so during energy needs, those triglycerides are broken down through a process called lipolysis. So that breaks down into a glycerol and a free fatty acid. So the white adipose tissue, as I mentioned, has these large unilocular lipid-filled vacuole cells. These are the adipocytes. But it also has vascular fraction, a stromal vascular fraction, where there are endothelial cells, there are pre-adipocytes, and then there are the supporting parasites, or the smooth muscle cells. There's also a resident population of inflammatory cells that really varies a lot depending on the person. Okay, so current tissue engineered models right now, they take either animal cell lines or they differentiate human adipose-derived stem cells. So the animal cell lines are often marine. They secrete only 1% to 2% of the leptin of mature adipocytes, and leptin is one of those key cytokines that's secreted by the adipose tissue. They often don't translate again to the human situation. Now, differentiation of human adipose-derived stem cells is gaining a lot more focus, but you can see right here, I think you can, they form these multilocular adipocytes, which is not characteristics of this large lipid-filled unilocular adipocyte. So this is all one lipid droplet as opposed to these multiple little ones. So you can tell that the phenotype of these models is really different than that that's actually experienced in vivo. So we thought that we needed to make some criteria for a successful white adipose tissue model. Obviously, first on our list was that we wanted the cells to remain viable. We wanted them to have that unilocular lipid morphology, which is really missing in a lot of the models. We wanted them to maintain similar number of cells, as well as triglycerides, which is what is actually in those unilocular lipid-filled vacuoles. We wanted them to maintain functionality, so the secretion of glycerol. We wanted there to be multiple cell types because there's an interaction between that stromal vascular fraction and actually those mature unilocular cells. We also want these to be sustained long-term. So this is really important when we're thinking about a disease model, because obesity actually takes months to develop and transition into an insulin-resistant type 2 diabetic phenotype. So this isn't going to happen overnight. So we're going to need a system that's going to be stable in culture for long term. We also wanted them to respond to different stimuli with a lipolytic response. Okay, so our hypothesis is that silk scaffolds will provide a 3D framework that will maintain those mature unilocular adipocytes ex vivo. And again, this is so that we can use this for a long-term culture model, as well as for eventually you can produce this as a place filler for lipoaspirin to be used in autologous soft tissue regeneration. So why silk? I don't know if everyone's familiar with silk as a protein, but it's got some really fantastic properties. You can see here, this is actually one of the cocoons from a Boxing mori worm, and so we can process this in an all aqueous format to get this like gel-like. It's very liquidy material. So an important thing is that it doesn't have any cell-specific signaling sites, so it's very inert, so you can control your environment. It's also cytocompatible. It can be formatted to various material formats, so we can make films, we can make sponges, you can do gels with it, you can electrospin it, it's very versatile. You can also change the mechanics and the degradation properties depending on how you actually process the silk, and I'll point out a few spots where that can be controlled. We've also done some bioactive functionalization in my lab where you add something like RGD, which is a cell binding sequence or motif, to the cell if that's required. It's also been stabilized with different compounds, so for drug delivery. So it's got these different characteristics that you can control and manipulate depending on what your purpose of your experiment is. So I'll just walk you through really briefly how we process these cocoons. So you get this cooked worm in a cocoon. You cut it up into different pieces, and we boil it in a sodium bicarb solution. And that's really because there's two proteins in the cocoons. There's sericin, which holds together the silk by bromine. And so in this step, we actually get rid of the sericin. This is also one of the steps where you can control the mechanical properties of the resulting solution. So if you were to boil it long term, you would actually decrease the molecular weight of the silk. So that would decrease the mechanical properties. Once you've done that, we dissolve it in lithium bromide. Then we put it in a dialysis cassette to get rid of the lithium bromide. And then you're resulted with this silk solution that you can centrifuge to get out any of the debris that you might have missed from the cocoons. So this step is usually where I will actually lyophilize the silk. It can be stored at 4 degrees C for maybe a month. But if you lyophilize it, it can stay indefinitely at room temperature. So then when I do want to make silk scaffolds, what I do is I dissolve the silk in H5P, and then we use salt as a porogen. So by sieving the salt, you can create different diameter sizes, and I actually use between 500 and 600 micron salt, which is actually quite large. Most of the other people in my lab use anywhere from 200 to 400 because they have smaller cells. But you saw the unilocular adipocytes that are large. They can be up to 100 microns themselves. So in order to actually get into the scaffold, we need to have larger pore size. Then once the silk penetrates the bottom of the vial, we let the H5P evaporate. And then we add methanol to induce beta sheets. So in silk, beta sheets kind of like cross-linking. It's actually how you make it so that it's not soluble in water anymore. And so this is one of the steps too where you can really control the mechanical properties as well as the degradation rate. Methanol is the available line. You can do water annealing, which would be not as many beta sheets, therefore it would dissolve much quicker. Or you can autoclave it, which creates more beta sheets, and then it would dissolve much quicker. Or it would dissolve. And at this point, we remove the methanol, and then we dialysize it again to remove all the salt, and then what you're left with is just that silk that's surrounding what used to be those salt pores. Okay, so what I'm going to concentrate on is using these silk scaffolds for adipose tissue, but I just wanted to put this slide in here because depending on the formatting, these silk scaffolds can be used for intestine. There's been a recent paper for cortical brain tissue or kidney or bone, so it really has a large diversity of mechanical properties and applications. Okay, so my approach is actually to take liquefied adipose tissue. So you can get that from lipoaspirin. which is just liposuction. Or what we often do is we'll get abdominoplasties, which are tummy tucks. So we'll actually just get like a chunk of tissue. We'll take off the skin, and we'll take off the scarpa fascia, and then I actually stick it in the blender, which sounds really gross, and it is. It actually ends up looking like nacho cheese, kind of that consistency and that color, which everyone always loves, right after lunch. You guys have some snacks, right? So yeah, so I soak the silk scaffolds. So I soak them in this liquefied adipose tissue. Then I can culture them for 4 days or 14 days or up to 3 months actually, so I'll show you some of that data. And what I used as a control was explants because that's the closest thing to the tissue that we can get. Okay, so again, we're going to go through the different criteria and how we tested them. So first we want to see if the cells were viable, and you can see here that the cells are alive and they're dispersed throughout the explant, so you can kind of see that they're in these different pores. There's some empty pores too, which is actually good so that there's actually perfusion of nutrients to the center. This is a scaffold and an explant that has been split in half. And you can see that the cells really are alive. And they seem to have that unilocular lipid morphology in the bright field, but we can't say for certain. So the next thing we wanted to test was whether they actually had that morphology that is really essential. So these are some images. These are at day 14. So you've got the explant and the scaffold here. So the fat is, again, that red color. Nuclei is dappy, it's blue. The actin is phalloidin, so that's in green. And the silk actually shows up in both the blue and the red channel. So that's the kind of purple color right here. So you can see that at day 14, the cells are actually growing along the scaffold as opposed to the overgrowth that actually occurred in the adipose tissue. And I think this is really because the adipose tissue kind of compacts, and then they just sort of form like a 2D surface almost on the top of it. So here you can see this is the adipose tissue rotated, mainly just because I think it's beautiful. And if that's the only thing you take out of my presentation, I'll be happy with that. The fat is in fact actually beautiful. But you can really see that they're forming right along those pores of the scaffold. Okay, so the next thing we wanted to make sure was just that they were maintaining that lipid area, which is that triglyceride unilocular filled vacuole. And you can see that there really isn't any change between the two, which is great. So next we wanted to test whether there were a similar number of cells and triglycerides. So here I'm showing pico green, which is for the DNA content. And really consistent with what we saw in the histology results. The scaffolds seem to maintain their cell number, whereas the explants are actually having a fibrotic response where they're overgrowing. When you see that we normalize it by the DNA content, the triglyceride content is very similar, so that's great. Next we wanted to look at functionality. So again, glycolysis is the breakdown of triglycerides into free fatty acids and glycerol, so we test glycerol. And you can see that they maintain secretion of glycerol in culture. So next we wanted to look at multiple cell types. And again, adipose tissue consists of that stromal vascular fraction besides those adipocytes. And so those are the pre-adipocytes, the endothelial cells, and then those supporting parasites or the vascular smooth muscle cells. And really this is because there's a lot of literature on how co-culture models are more accurate because there's a lot of the parent signaling between cells. So this is really important for an accurate tissue engineered model. So PREF1 is the pre-adipocyte marker, and you can see that they are in the scaffold at day 14, which is great. CD31 is an endothelial marker, and you can see that it's actually forming almost right around the scaffold borders. It wasn't always this beautiful in all of the cultures, but there was always endothelial cells present. And then you can also see the same sort of thing happening with the parasites or the vascular smooth muscle cells. Okay, so next we wanted to see if these were actually sustained long term. And so again, this is for those chronic models where we think that diseases are going to need long term in culture to actually recapitulate the disease processing. So explants deteriorate in culture. I don't know, I'm sure you guys probably don't feel fat very often, but it's not very stable on its own. So they usually lasted to about 14 days, which is why I chose that time point for comparisons with the explants. But the silk remained intact, and we actually saw similar numbers of cells and triglycerides, which is great. So it's a stable culture system, which is what we need to actually perturb the system. So finally, we want to look at lipolytic responses. So is this actually responding to stimulation? And so epinephrine is a really known cause of lipolysis. So we saw this increase in epinephrine that was kind of dose dependent. When we tested 5 different patient samples, and we saw that this was a very consistent response, that it responded to epinephrine with an increase in lipolysis. The interesting thing that I think happened is that the variability between the patients and their responsiveness to the epinephrine is really inconsistent. They all responded, but not in the same manner. And so next we wanted to look at a drug treatment. So ACAR is a drug that is known as exercise mimetic, or an exercise pill. It actually causes secretion of glycerol or lipolysis, but it's really inconsistent in the literature. So this is something that they tried to get on the market, but it hasn't had consistent results. So the first patient we tested, and isn't this true of all science, worked perfectly, or it doesn't work at all, one or the other, but then it never works again. So our first patient looked beautiful. I had a dose-dependent response. And then we decided to test 10 more patients. So I think this is fairly interesting because we only saw 3 patients that actually had this response with lipolysis. And so we thought maybe it was BMI related, so we plotted it against BMI, which is the only indicator we have right now with the samples that I get. And so it's obviously not related to BMI. And this is probably why all of the clinical trials with it were so inconsistent, because it's very patient specific. And that could be related to, the fitness level or the metabolic status or some other unknown factors. Okay, so just a summary of the system. The cells were viable and they were distributed throughout those scaffolds. The unimolecular lipid morphology was maintained, which is consistent with its in vivo phenotype. We maintained cell numbers and triglycerides, as well as functionality, which was that glycerol secretion. And the system supported multiple cell types, so it's likely to be more physiologically relevant than these stem cell differentiated models. These were sustained in long-term cultures, so it suggests that we could use these for a chronic model. And then we also recapitulated lipolytic responses with epinephrine in the system and used it as a treatment for obesity to show that there is actually a lot of patient variability. So silk bio as a biomaterial for this purpose enabled a long-term tissue engineered model, so this could be used to study disease progression and for testing potential therapies. This is also good for soft tissue regenerative therapies where you need it to slowly degrade in culture, but maintain that adipose tissue. Okay, so now I'd like to just go into some future research directions. So using this tissue engineered model to study disease mechanisms. What I mentioned before is that the obesity is really an ever-growing problem. I think everyone would know that. That the world is getting fatter, but Colorado is a nice state amongst some darker states around it, so that's good. But even here, you're getting an increase in obesity as well. So the problem isn't really that people are obese, it's that this obesity is actually linked to so many different diseases. And in particular, what I'm going to talk about is type 2 diabetes. So obesity is a well-established risk factor for type 2 diabetes. And type 2 diabetes is the 7th leading cause of death in the US. And as you can imagine, as this obese population is increasing, the amount of type 2 diabetic patients will also increase. And with no suitable clinical options today, there's really a need to determine what is actually causing this change. The problem is that that link is really unknown. So is it a risk factor or is it a cause? So there's a lot of different hypotheses. So one of them being, and one of the more predominant ones, is that it's related to chronic inflammation. So as your adipose tissue is expanding, you actually cause systemic chronic inflammation in your body, which is going to affect all those other tissues. There's likely to be some population variability. We even saw that in our model. And whether that's related to age, sex, fitness level, you could go on and on. As much information as you can get from these patients would be crucial. The interesting one that I've been reading more about, I don't know if you guys have as well, is the exposure to environmental factors. So there's these things called obesogens, and I swear I didn't make that word up. It's actually a word. And there are things like BPA or nicotine. that when the parents or the mother is actually exposed to them, the baby in utero, to these different chemicals, it predisposes them to be obese later in life. So there was these beautiful set of experiments where they took mice that were pregnant and exposed them to some of these chemicals, and then exposed the offspring to the same diet, so with or without the chemical. And the ones with the chemical actually became obese over the ones that weren't exposed to the chemical. And what's even scarier about that is that the offspring of those pups also had effects too, although not as much. So it wasn't just one generation, it was two. So I think that's a really interesting area of research that we should learn more about. Another thing is the paracrine signaling with other tissues. So again, because this is an organ, an endocrine organ that secretes different chemicals, how are these affecting other tissues and how is that crosstalk influencing this transition from obesity to an insulin-resistant type 2 diabetic state? And finally, there's definitely some genetic predisposition and many other factors that we're only learning about more recently. Okay, so my kind of main goals that I see in my 5-year vision would be developing a 3D adipose co-culture model to be used as a screening tool. And then what I'd like to do in the second project is use that tool to actually look at targeting key receptor mediated pathways. And I'll go into more details about that in the next slide. Some other broader visions that I could see from my research would be to explore some of these environmental factors underlying the mechanism of type 2 diabetes, those obesogens, as well as for the fourth project, studying the effect of paracrine signals with some of these other tissues that are really active in type 2 diabetes. So this is kind of an overall conceptual model. So basically we'll target different pathways suspected to play a role in obesity. We'll test the mechanisms ex vivo screening. We'll identify some candidate targets. This is likely to be a feedback loop where the ultimate goal is to identify key cells and components that promote insulin resistance. On the flip side of that would also be to stimulate these with obesogens or interface with other tissues. Again, identify candidate targets for the transition to that systemic insulin resistance, which will be a feedback loop, I'm sure, as well. So the ultimate goal being here that we'd really like to create some therapeutic targets. Okay, so this is kind of how I envision the first project. So you get cells from different patients and you have some markers. You have BMI, you'd also have diabetic status, age, sex. You'd stimulate them with different compounds that are suspected to initiate diabetes. And then you would look at glucose uptake. So that's insulin-stimulated glucose uptake is really the marker of an insulin-resistant phenotype when it starts to decrease. And also you'd want to look at phosphoproteins, so the phosphorylated pathways that are being initiated by these different stimuli and in relationship to these different human components. So in the second part, it would be to computationally model this interaction between the inputs and the outputs. So on the last project, the focus would kind of be the NIH and NIDDK, I think, for these sort of grants. Whereas this one, I think you could get some of these more NSF grant opportunities, and there are a few actually NIH opportunities as well. So this is kind of the matrix that I would envision, where you have the different patient samples and the treatments, you have your phosphoproteins, and then you have this output, which would be your glucose uptake. And you could use this to actually look at metabolic signaling cascades and the metabolic markers of disease. So for the third project, it would be to create a new model where we would use embryonic stem cells to create more of that in utero environment. You differentiate the stem cells, expose them to some of these different chemical exposures, and then you'd actually treat them with free fatty acids, glucose, insulin, and see how they respond. For paracrine signaling, I could envision having a perfusion system where you would interface, for instance, adipose tissue and skeletal muscle tissue, expose them to different factors and see how they respond individually and collaboratively to see if we can get some of that crosstalk between the two tissues. Okay, so other potential applications of interest that I could see would also be to create other adipose depots, so that I've been talking about subcutaneous adipose tissue throughout. There's also some less studied adipose depots, for instance, the brown adipose tissue, mammary tissue, or bone marrow. I'd also see this really being interfaced in a full thickness skin model, especially because diabetes affects the healing process. So if you had a diabetic adipose model that's with the epidermis and the dermis, you could look at how that affects the healing process. I think another really interesting area would be to collaborate with someone on incorporating cancer cells, because I think this would be a really interesting model to study cancer progression, in particular in that mammary depot, so you can do breast cancer. So I was looking up everyone at CSU and I saw some potential collaborations, in particular lab on the chip systems, the computational modeling and machine learning I think would be really interesting with different inputs and outputs, as well as looking at some different environmental pollutants. Okay, so I talked about the perils of the pot belly, and I've only really focused on type 2 diabetes, but the amazing thing about obesity, or not so amazing thing depending on how you look at it, is that it really could result in a lot of different research opportunities that are funded by a lot of different institutes. So I have had a small amount of grant writing experience. So I did have an F-32 that scored really well, and they actually told me it was going to be funded. But then it was when sequestration happened, and so I've already felt the heartbreak of grant writing, having one taken away. But I've most recently been working on my own American Diabetes Association grant, and so that's an initiator grant, and you can only have one per school, and they selected me, so I'm going to be submitting that soon. I just wrote a grant with my boss for NSF, which was very different than the NIH grant writing process, so that was a really good learning experience. And that was actually on doing cultured meat, which was a little weird because all of the preliminary data was human, but we did propose bovine, so it's okay. I also had an opportunity to work on a consortium grant, which is another learning experience as well, because it was with two different other collaborators. And it scored really well, so I don't know how we'll do on that one. Okay, so I'm right on time too. So the last 10 minutes I'd just like to talk about my teaching interests, and it's something I'm really passionate about. So I thought I'd first go through what I think is important, then go through some of the classes that maybe I could teach here. So my teaching approach is really to try to get students actively involved in reading and or doing current research, because I think if you see the current problems that engineers are facing and different people in the literature are doing, it makes it become more alive for you. The same idea, incorporating technology into the classroom, was really something that I valued a lot at RPI. We worked with MATLAB and Console and other modeling techniques that really I felt like prepared me to go out and to actually use those in an industry-related job. And I think the overall goals are really that I think that you need to engage the students and get them excited. So if they're excited about the material, they're going to go through the hard work of learning it. And I think that goes hand in hand with actually portraying enthusiasm as a teacher for the subject matter and making it come to life for them. So I think also an important thing is having the students try to test new hypotheses and come up with new questions to understand a problem, and I think that also goes hand in hand with them actually questioning the facts and thinking about the big picture so that they can relate those concepts actually to their everyday life and value the importance of what they're actually learning. I think whenever